Hey everybody, welcome back to another video. Today we're going to be talking about ion channel dynamics and how they influence action potentials and spike timing dependent plasticity. We're also going to have some focus on how the timing of EPSPs, or excitatory postsynaptic potentials, affect the calcium influx during LTD, and how dysfunction in specific channels like KCNQ2/3 contributes to neural circuit abnormalities, like, for example, in tinnitus or hyperacusis. Now, I haven't made a video in my Foundations of Neuroscience lecture series yet on the action potential, so here's going to be a quick rundown. Now, the action potential, or AP, is a rapid, all or none, so to speak, electrical impulse that neurons use to send signals. Now, its shape and durations are determined by the coordinated opening and closing of specific ion channels, like we discussed in the lecture series. Now, the three main ion channels, or the ion types, are involved, which is sodium, or Na+, which initiate the spike. Potassium, or K+, channels repolarize and hyperpolarize the membrane, which means bringing the membrane potential down. And chloride, or Cl-, channels, they contribute to stabilizing the membrane potential, usually via inhibition, or during the release of the inhibitory neurotransmitter, GABA. Now, together, these channels create the phases of the action potential, which is like an upstroke or like an increase of a graph, as you can see on the image above, which is called the depolarization, a downstroke, which is called the repolarization, and a after hyperpolarization or hyperpolarization phase. Now, voltage-gated sodium, or Na+ channels, are responsible for the initiation, the rising phase of the action potential. And when the neuron's membrane is sufficiently depolarized to reach a certain threshold, which is around about minus 55 millivolts, a large number of sodium channels open rapidly, and the sodium ions rush into the cell, down their electrochemical gradient, like we discussed in the lectures, carrying a positive charge. And this influx causes a fast depolarization of the membrane, making the interior more positive. So basically, Na+ or sodium channels kind of ignite the spike. They generate the steep upwards, or the steep upswing of the action potential. And at the peak of the action potential, which is often around plus 30 or plus 40 millivolts, the sodium channels begin to inactivate, or they stop conducting, even though the membrane is still depolarized. Now, the inactivation is really important for stopping the depolarization phase and initiating the next phase of the action potential. Now, the rapid opening and the subsequent inactivation of the sodium channels, they underlie the brief duration of the spike, and they also contribute to the absolute refractory period, which is the brief interval during which the second action potential cannot be initiated. And without functional sodium channels, neurons, of course, cannot fire typical action potentials. Now, following the sodium-driven depolarization, the potassium, or the K+ channels, take the center stage, so to speak, to repolarize the membrane. A fraction of voltage-gated potassium channels opens more slowly during the spike, often as sodium channels are inactivating. Now, because potassium is more concentrated inside the neuron, opening the potassium channels let the K+ ions flow out, carrying positive charge out of the cell. And this outward current counteracts the sodium-induced depolarization and drives the membrane potential back down, the falling phase on the graph. Now, the efflux, which is the opposite of influx, of the K+, rapidly brings the membrane potential towards the K+ equilibrium potential, which is negative, around minus 90 millivolts. And in fact, the K+ currents typically overshoot the resting potential, causing the membrane to become transiently more negative than the rest. This is the hyperpolarization, or the after hyperpolarization phase, or the undershoot phase, basically. Now, the after hyperpolarization occurs partly because some voltage-dependent potassium channels are slow to close. They continue to let the potassium out even after the membrane has repolarized. Now, additionally, certain potassium channels, like, for example, calcium-activated potassium channels like BK or SK channels, they may open during the spike due to the rise of intracellular calcium, which contributes to a longer-lasting hyperpolarizing tail. Now, the AHP is essentially a hyperpolarization that follows the spike and raises the threshold for a short time. Now, this corresponds to the relative refractory period that we discussed previously, during which a stronger stimulus is needed to fire another spike. Now, by hyperpolarizing the membrane, K+ or potassium channel activity limits the firing rate, and it helps neurons to reset after each spike. Now, neurons often express multiple subtypes of potassium channels, which each contribute to different phases of repolarization. For example, the delayed rectifier K+ channels activate during the spike to repolarize it, while the M-type K+ channels, like, for example, KCNQ2/3, which some of you might recognize from Tzounopoulos' paper, provide the slower, non-inactivating K+ current that basically governs the medium after hyperpolarization and excitability between spikes. And this is really important, especially in tinnitus, because with a decrease in KCNQ2/3 activity, this of course increases the excitability between spikes, leading to another action potential potentially being fired when it's not supposed to. Now, the diversity of the K+ channels ensures that even if some inactivate or are blocked, others can repolarize the membrane, basically making repolarization a really robust process. Now, let's talk a little bit about chloride channels. Now, the chloride or Cl- channels are not primary drivers of an action potential. They play a significant role in stabilizing the membrane potential and counteracting excitation. Now, the chloride equilibrium potential, or E_Cl, in mature neurons is usually near or slightly below the resting potential. Now, when Cl- permeable channels, often activated by inhibitory neurotransmitters like GABA or glycine, open, Cl- ions often flow into the neuron, since intracellular Cl- is kept low. And this influx of calcium carries the negative charge in, causing hyperpolarization, making the interior more negative, or clamping the voltage near the chloride equilibrium potential. Now, during an action potential, chloride currents can modulate the shape of a threshold. For instance, if chlorium channels are open, they oppose the depolarization, as the chlorium influx or the K+ efflux both drive the membrane negative. In fact, voltage-gated Cl- or chlorium channels exist in some cells, and along with K+ channels, they contribute to the late hyperpolarization phase. Now, the combined effect of K+ efflux and Cl- influx is what generates the hyperpolarizing after potential that follows a spike sometimes. Now, thus, while Cl- channels do not create the upstroke of AP, or the action potential, they are one of the key components in shaping the neuron's return to a polarized resting state and inhibiting excessive excitability, of course. Now, clinically, if chlorium regulation is distributed, or in other words, if the chlorium gradient is reduced such that the equilibrium potential is less negative, inhibitory signals often become weaker, which is interesting, illustrating how important the chloride currents are for normal neural firing dynamics. Now, a little reminder to those who don't know. Now, neurons integrate synaptic inputs on their dendrites and their soma, and the timing of those inputs relative to the neuron's own firing is critical for synaptic plasticity. And in spike timing dependent plasticity, or STDP, the relative timing of a presynaptic spike, which causes an excitatory postsynaptic potential, or EPSP, and a postsynaptic spike determine whether the synapse is strengthened or weakened. Now, generally speaking, if a presynaptic EPSP arrives before the postsynaptic neuron fires, it contributes to firing and is followed by a postsynaptic depolarization. Now, this tends to, of course, induce long-term potentiation. polymerization or synaptic strengthening. Now, if an EPSP arrives after the postsynaptic neuron has fired, or in other words, the neuron was recently active and is now in a hyperpolarized state, the opposite happens. It tends to induce long-term depression or synaptic weakening. Now, this rule can be summarized as pre before post equals LTP and post before pre equals LTD. Now, on the image above, you can see the typical STDP timing curve, which shows the synaptic change versus the interval between a presynaptic spike or a postsynaptic spike. And in bimodal or bisensory stimulation, this is called the bimodal interval. Now, when a presynaptic spike precedes a postsynaptic spike by a few milliseconds, or the positive T, as you can see, LTP occurs, the upward percentage change. Now, on the other hand, if a presynaptic spike arrives shortly after a postsynaptic spike, or the negative T, as you can see, the synapse undergoes LTD, or the downward change. And the time window is narrow. It's on the order of like maybe tens of milliseconds, if not less. And it transitions sharply from potentiation to depression on the graph. Synapses basically effectively detect the causal timings. If an EPSP arrives during the postsynaptic neuron's hyperpolarized or after the spike phase, it tends to weaken the connection due to the very minor and maybe prolonged low calcium influx. Now, to know more about calcium influx, you can watch one of my previous videos, but I'm going to give a small explanation now. Now, why does an EPSP arriving during a hyperpolarized phase lead to LTD? Now, the key lies in how the postsynaptic neuron state affect the calcium influx, of course, which is the trigger for plasticity. Now, after a neuron fires, it enters a after hyperpolarized state due to the K+ currents, as we discussed previously. Now, in this state, the membrane potential is more negative than usual, and if an EPSP, which is caused by a presynaptic glutamate release, arrives during this time, the neuron is less excitable. The EPSP will be smaller, since the driving force on sodium is reduced at the very hyperpolarized voltages, and more charge is required to reach the spike threshold. Now, the postsynaptic neuron likely won't fire again immediately from this EPSP, and crucially, the NMDA receptors that mediate the calcium entry are voltage dependent. This is very important. And during the hyperpolarized AHP, or as you can see this little section on the graph where the membrane potential is more negative, the NMDA receptors remain magnesium blocked or less activated. Now, thus, the pairing of the post-spike then EPSP yields only a minimal calcium influx, which is just enough to trigger LTD, but not enough to trigger LTP. Now, of course, experiments have shown that when a back-propagating action potential in the dendrite is followed by an EPSP in post before pre-order, the resultant calcium transient is smaller compared to when the EPSP comes first. Now, this smaller calcium signal preferentially engages the phosphatases, or the enzymes that lead to synaptic depression, rather than kinases, which leads to the strengthening. In other words, an EPSP arriving during the after hyperpolarization provides a weakly coincidence signal. It finds the postsynaptic cell in a refractory, recently active state, and thus the synapse is downregulated, as if the neuron post-dicted that the input was not casual for its firing. Now, another way to put it is in terms of spike pairing intervals. If the presynaptic spike lags behind the postsynaptic spike, or a negative timing interval, the synaptic weight is weakened. Now, the window for LTD is usually a bit wider and shifted compared to LTP. So, for example, in many cortical neurons, an EPSP that arrives up to minus 20 to 50 milliseconds after a postsynaptic spike can induce LTD, interestingly enough. And the most effective LTD often occurs when the delay is short. In other words, from 5 to maybe 20 milliseconds post-synaptic spike, which, it's different depending on what neurons you're analyzing. Now, in this scenario, the postsynaptic cell was just active, likely due to other inputs, or like in the Susan Short device, the auditory stimulation, for example. So, a lone EPSP arriving afterwards is interpreted as an unimportant or out of sync signal, which results in depression. Now, this timing-dependent LTD is thought to help neurons refine their timing relationships, or inputs that consistently fire after the neuron will lose influence, which prevents reinforcement of inputs that are not causal. Now, biophysically, as we noted before, the depressed calcium 2+ signal is the trigger. And studies using direct calcium uncaging have found that a weak, slow rise in calcium triggers LTD, whereas a large, rapid calcium influx triggers LTP. And so the precise timing during the hyperpolarized phase, of course, produces the former, which is the small calcium influx leading to LTD. Now, the induction of LTP or LTD at synapses is often described by the calcium control hypothesis. Now, according to this idea, the level and timing of postsynaptic calcium, or Ca2+, rise determines the direction of plasticity. Now, high amplitude, rapid calcium influx triggers LTP, like I mentioned before, and smaller or more prolonged calcium triggers LTD. Now, this makes intuitive sense. A big surge of calcium, for example, due to nearly coincident pre- and postsynaptic firing, activates calcium or calmodulin-dependent protein kinase 2 and other pathways that strengthen the synapses or strengthen the scaffolding proteins, while a modest calcium elevation favors the protein phosphatases, like calcineurin, that weaken the synapse or they weaken the scaffolding proteins. But what determines how much calcium enters? This is where the ion channel kinetics or the timing of channel opening and closing come into play. Now, there's different calcium sources, of course. Now, during an EPSP spike sequence, calcium enters a dendritic spine primarily through the NMDA receptors, which require depolarization to relieve the magnesium block, and through voltage-gated calcium channels, or VGCCs, that open during depolarization, for instance, when the action potential back propagates into the dendrite. Now, the amount of calcium entry is steeply nonlinear with voltage. If the postsynaptic membrane is strongly depolarized, in other words, by an EPSP that coincides with an action potential, the NMDA channels fully conduct and VGCCs open broadly, yielding a massive calcium transient. And if the depolarization is weak or brief, the calcium entry is limited. Now, if we talk about LTP versus LTD calcium thresholds, some empirical studies with the calcium uncaging in the spines, like I mentioned before, have found a threshold calcium concentration above which LTP is induced, and a lower range that induces LTD. For example, one classic result, injecting a high calcium transient, around 5 micromolars, into a CA1 pyramid cell spine led to LTP, whereas a smaller elevation, or around 1 micromolar or less, produced LTD. Now, in STDP, a pre-before-post pairing, the EPSP happens, then the postsynaptic spike, produces a large, rapid calcium rise that crosses the LTP threshold, whereas a post-before-pre-pairing yields a smaller calcium rise that only meets the LTD threshold. Now, notably, blocking the calcium rise or clamping it can abolish these plasticity changes, which really underscores that calcium is the signal for synaptic change. Now let's talk about the action potential width and calcium entry. Now, the duration of the postsynaptic spike, or the action potential, can influence calcium influx. A broader action potential keeps the membrane depolarized longer, which allows more calcium to enter through the VGCCs and NMDA receptors. Now, intuitively, a longer depolarization might be expected to favor LTP by increasing calcium. However, interestingly, if an action potential is excessively broadened, for example, by blocking certain potassium channels, it can sometimes enhance LTD under STDP protocols. Now, how does this happen? One explanation is that while the total calcium is higher, its kinetics may shift such that it preferentially activates slow calcium-dependent phosphatases. Alternatively, prolonged depolarization might trigger homeostatic mechanisms or different receptor activation. In general, though, narrow, sharp spikes, as normally shaped by fast potassium currents, tend to confine calcium influx to a brief spike. Now, slower repolarization due to altered K plus channel activity can increase calcium entry, potentially raising the likelihood of hitting LTP thresholds, but also possibly altering which enzymes respond. And as we know, KCNQ 2/3 channels are disrupted in people who have tinnitus. If you can take a look at the illustration above from Tzounopoulos' paper. Now, the after hyperpolarization and the timing of depolarization, like we discussed in part 2, a strong AHP or after hyperpolarization driven by potassium or K plus channels will suppress immediate redepolarization. If the AHP is large, a subsequent EPSP during that period results in a smaller depolarization and thus smaller calcium entry, biasing towards LTD. And this is really important. Now, on the other hand, if K plus channel activity is reduced, for example, a smaller AHP, the neuron's membrane doesn't hyperpolarize as much after the spike. An EPSP arriving shortly after might produce a larger depolarization, perhaps even trigger another spike or a dendritic spike, and thus a larger calcium transient, shifting the outcome more towards LTP. Now, in this way, the potassium channel kinetics set a voltage environment, so to speak, that either raises or lowers the bar for an EPSP to cause significant calcium influx. For example, M-type K plus channels like KCNQ 2/3 provide a persistent outward current that hyperpolarizes or stabilizes the membrane. If they deactivate or if they are dysfunctional, the neuron becomes more excitable, and EPSPs can, of course, summate more, potentially yielding higher calcium spikes and favoring LTP. Now, HCN channels, like I mentioned in my previous video, they help bring the neuron back to its resting membrane potential, meaning they kind of help the hyperpolarization or the AHP or the after hyperpolarization phase to be shorter. And because of this, combined together with the reduced KCNQ 2/3, it means that the neuron in general becomes more excitable because the outward current or the efflux of potassium is dysfunctional, meaning that the membrane actually stays more positive due to this. And of course, this is reinforced, or I meant that the dysfunctional potassium efflux or K plus efflux is actually reinforced by the normal functionality of the HCN channels, leading to more excitability. Now, if we talk about the timing of NMDA versus AMPA currents, fast AMPA receptor currents depolarize first, and slower NMDA currents follow, admitting calcium. Now, if voltage-gated potassium channels activate too quickly, they can curtail the depolarization that would otherwise unblock NMDA receptors. Thus, fast repolarization by potassium can limit the window of NMDA calcium entry. Alternatively, if repolarization is slightly delayed within a certain window, it may allow more NMDA current. There is a delicate interplay, so to speak. The neuron needs to repolarize to end the spike, but the precise timing and rate of repolarization set by channels like the KV3, KV4, KCNQ, etc., will of course fine-tune the calcium signal. Now, biochemically, CAMK2 versus calcineurin exemplify the different calcium thresholds. CAMK2, which is a kinase necessary for LTP, has a relatively high calcium/calmodulin affinity. When calcium spikes are high and brief, it can undergo autophosphorylation to stay active after a big burst of calcium. In contrast, calcineurin, which is a phosphatase that promotes LTD, can be activated by lower sustained calcium. So basically, high-frequency bursts or strong coincidences, which produce repetitive spikes and summated EPSPs, generate high-frequency calcium spikes that tip the balance to CAMK2 and LTP. And of course, on the other hand, low-frequency activity or late EPSPs produced smaller calcium rises that preferentially activate calcineurin PP1, for example, yielding LTD. Now, ion channels also modulate spike frequency and bursting, which indirectly affect calcium accumulation. If a neuron fires a burst of action potentials due to certain ion channel properties, the cumulative calcium from multiple spikes can reach LTP range, even if single spike calcium might not. And on the other hand, if channels enforce only single spikes with adaptation, the calcium may remain in the LTD range unless a very coincident EPSP occurs. Now, among the many K+ channels or potassium voltage-gated potassium channel subtypes, KCNQ2/3 channels, which are also known as KV7.2/7.3 or M-type K+ channels, play a really important role in controlling the, of course, neuronal excitability. Now, these channels generate the M current, a non-inactivating, slowly closing potassium current that is active at subthreshold voltages. In practical terms, basically a KCNQ2/3 channels are partially open at rest and during small depolarizations, providing a braking influence on the neuron. And they oppose depolarizing inputs and stabilize the membrane potential, making neurons less likely to fire spontaneously. Now, this has, of course, several important consequences. First one is, of course, regulating excitability. KCNQ2/3 channels, they set the threshold and responsiveness of neurons. And if these channels are very active, a neuron requires a stronger excitatory input to reach threshold because the M current is pulling the voltage down or preventing rapid depolarization. If the M current is suppressed, for example, by muscarinic acetylcholine receptors, which is how they got the name M channels, the neuron becomes much more excitable and prone to repetitive firing. Indeed, loss of function mutations in KCNQ2 or KCNQ3 cause hyperexcitability, like, for example, in epilepsy, because neurons fire too easily without stabilizing M current, or like, according to Tzounopoulos' research, tinnitus. Now, the gain of function mutations, or drugs that open the KCNQ2 channels, dampen excitability and can even cause hypoexcitability in some cells, like, for example, Trobalt, or it's also called reticabine. Now, the KCNQ2/3 basically are the volume knobs on excitability. Turning them up quiets the neuron, so to speak, and turning them down makes the neuron louder, I guess. Now, if we talk about the medium after hyperpolarization, or the MAHP, the KCNQ2 channels contribute to the medium duration after hyperpolarization following a burst of spikes. When a neuron fires a train of action potentials, calcium-activated K+ channels, which we'll call SK, produce a fast AHP. And KCNQ channels, along with the HCN channels, produce a slightly slower AHP, or after hyperpolarization, that can last hundreds of milliseconds. This MAHP helps limit repetitive firing by keeping the neuron hyperpolarized between spikes. Now, in some experiments in mice lacking the KCNQ2, in cortical pyramid cells, for example, the medium HP is greatly reduced and an abnormal afterpolarization appears instead, leading to more spikes per stimulus and higher firing frequencies. In other words, without KCNQ2, neurons don't pump the brakes after firing. They can enter high-frequency firing or burst more readily. Now, experimental block of KCNQ channels with inhibitors like XE991, they can likewise cause neurons to fire high-frequency bursts instead of single spikes and reduce spike frequency adaptation. This basically shows that KCNQ2/3, they act to, I guess, enhance the reliability and prevent the runaway excitation, which coincidentally is exactly what we need with tinnitus. Now, if we talk about firing reliability and precision, especially if we're talking about pathways that require high-frequency signaling, like the auditory pathway, KCNQ2 channels actually help maintain consistent spike timing. A really good example comes from the auditory brainstem synapses, for example, the calyx of health synapses. Now, some research shows that during very fast firing, or hundreds of hertz, KCNQ channels activate cumulatively and prevent the axon terminal from depolarizing excessively, which in turn prevents the sodium channel inactivation and keeps the action potential waveform stable. And by holding the baseline potential a bit lower between spikes, KCNQ channels ensure that each spike invades the terminal with full amplitude and precise timing. Now, when KCNQ is blocked, the presynaptic spikes start to fail or they start to wobble, and the sodium channels inactivate more. The action potentials broaden, and the synaptic transmission becomes unreliable at high frequencies. Yeah, you heard that? Now, basically, KCNQ channels paradoxically facilitate high-frequency transmission, not by making the neuron fire, but by making sure it doesn't overfire or depolarize in an uncontrolled way. And in general neural coding, this basically means that neurons with functional KCNQ channels can fire repetitively with less jitter. And each interspike interval is more consistent because the M current is buffering the membrane potential. This is why you can often see KCNQ described as preventing hyperexcitability, but also maintaining the fidelity of spike trains. Now, if we talk about threshold and subthreshold oscillations, KCNQ channels, they also influence the resting membrane potential and threshold. They are active at rest, so they contribute to the resting conductance. And neurons that have resonance or subthreshold oscillations, like, for example, certain thalamic or cortical neurons, rely on an interplay of KCNQ and HCN currents. KCNQ, which is the hyperpolarizing current, like I mentioned before, and the HCN, which is the depolarizing cation current, they together form a brake and accelerator. A brake and accelerator system that can, for example, stabilize firing patterns. For example, you can see that blocking KCNQ not only increases the firing rate, but can induce oscillatory bursting because the neuron no longer has the stabilizing potassium current to counterbalance the inward currents. Now, from a plasticity standpoint, KCNQ's 2/3's effect on excitability will also influence STDP outcomes. Now, if KCNQ channels are very active, a given pairing of pre-post might not yield as large a depolarization or calcium influx, possibly biasing towards LTD or no change, because the neuron is harder to excite. And if KCNQ currents are suppressed, for example, I don't know, by neuromodulators like acetylcholine during attention, neurons become more responsive, and the same pairing could produce a larger postsynaptic depolarization and more calcium, thus favoring LTP. Now, in this way, KCNQ 2/3 can set a gain on plasticity indirectly by modulating how easily postsynaptic spikes coincide with EPSPs. So this is the example that I gave about acetylcholine during paying attention is, of course, one of the reasons why people remember stuff better while paying attention. That's why you should always pay attention in class, kids. Now, dysfunction in potassium channels can very profoundly disrupt the balance of excitation and inhibition in neural circuits like we just discussed, sometimes leading to maladaptive plasticity, or the brain reorganizing itself in such a way that does not benefit the person. Now, a really good example, of course, is tinnitus. And according to Tzounopoulos' research involving the KV7 or KCNQ channels, is a really key player in this process. Now, the dorsal cochlear nucleus, which is the brainstem, I guess, center implicated in tinnitus, watch my previous videos. The principal neurons called the fusiform cells, they normally receive a mix of auditory nerve and inhibitory input. Now, they exhibit STDP based on combined auditory and somatosensory signals. In noise-induced tinnitus models, it was found that fusiform cells become hyperactive or increased spontaneous firing, and their synaptic plasticity rules shift. Tzounopoulos and his colleagues discovered that this is tied to a reduction in KCNQ 2/3 channel activity in these neurons. Now, normally after noise exposure, some animals' fusiform cells compensated by upregulating the KCNQ currents and downregulating the HCN currents to keep excitability in check. But in mice that develop tinnitus, this homeostatic compensation fails. The KCNQ 2/3 currents remain suppressed, leaving the cells hyperexcitable. Basically, tinnitus that arises in these cases where the K plus break is lost and neurons start firing too much on their own. Now, what does this mean for STDP? Now, it turns out that STDP curve in fusiform cells is dramatically altered in tinnitus. Now, in normal conditions or in noise-exposed animals that did not develop tinnitus, pairing inputs in a certain order might produce mostly LTD, which is thought to, of course, be protective or adaptively filtering inputs. Now, however, in tinnitus-afflicted animals, the same pairing produce exaggerated LTP. Specifically, recordings show that tinnitus model fusiform cells have an inverted and upward shifted STDP rule. More LTP across various timing intervals and much less LTD, unfortunately. And conversely, the resilient mice, which were exposed to loud noise but no tinnitus, showed a bias towards LTD in the same paradigm. Now, this inverted STDP in tinnitus means that inputs that are slightly mistimed may still potentiate. And one can, of course, imagine that normally, if an auditory signal arrives without proper context, the synapse might weaken, preventing noise from being encoded as a signal. But if K plus channel dysfunction removes the constraint, those inputs may potentiate instead, reinforcing a spurious signal, possibly contributing to the persistent, phantom firing. Now, mechanistically, the loss of KCNQ 2/3 in fusiform cells means a reduced AHP and increased excitability, just like we described in part 4. And the fusiform cell may fire more easily and may also back-propagate spikes more strongly into its dendrites. So when somatosensory and auditory inputs coincide, which is how the DCN fusiform plasticity is driven, the postsynaptic cell might now generate a larger depolarization and calcium influx than it normally would. And this could push calcium signals into the LTP range for nearly all pairings. Additionally, if the neuron is spontaneously firing due to hyperexcitability, the presynaptic inputs that happen to follow its spikes, which should induce LTD, might not actually depress because the ongoing firing and depolarization keep the calcium levels somewhat elevated. Now, essentially, the calcium threshold for LTP is more easily exceeded when the KCNQ is down. Everything the cell, quote-unquote, hears might cause a strengthening, which is basically a recipe for runaway potentiation and persistent activity. So put it all together basically means easier spikes. Now in Tzounopoulos' 2013 study, it provided the direct evidence. It's the study from where I got this graph from above. A reduction in KV7.2/3 or KCNQ 2/3 activity was necessary for tinnitus induction, and preventing that reduction pharmacologically could prevent tinnitus. And they showed that after a noise trauma, only the mice that developed tinnitus had a positive shift in KCNQ channel voltage activation. which basically means the channels were harder to open, effectively a loss of function. And by administering a KCNQ opener that shifted the channel activation back to negative, making the channels easier to open, they prevented the development of tinnitus in those animals. And this highlights that it was indeed the K plus channel plasticity, or pathogenic plasticity as they called it, that allowed the aberrant STDP and hyperactivity to take hold. And in the framework of STDP, restoring KCNQ function likely restored the normal LTD or LTP balance, which ensures that only appropriately timed inputs induce LTP, while others cause LTD or no change. Now, if we connect the dots, the potassium or K plus channel dysfunction can dysregulate STDP by altering the neuronal excitability and the resultant calcium signaling. And in tinnitus, the lack of KCNQ-mediated hyperpolarization meant that all incoming activity tended to strengthen the synapses. The timing rule flipped to favor potentiation, basically. Now, this led to reinforcing circuits that probably encode the phantom spiking, or the fusiform cells start firing as if there's always input. And it's a case where a normally adaptive plasticity rule becomes maladaptive. Now, the condition of the neuron, which is set by the ion channels, biased the synaptic plasticity towards a pathological outcome. Now, it's really important that I mention that enhancing, of course, the KCNQ currents after tinnitus onset has been explored as a therapy. The idea being to reintroduce the brake that allows the brain to unlearn, quote unquote, the phantom signal through restored LTD. And of course, some researchers, they have suggested KCNQ channel activators as maybe a precision treatment to induce beneficial corrective plasticity in tinnitus patients. Unfortunately, Thanos' trials failed due to his molecule being insufficient, but I believe that there are different ways that we can mediate or we can change this KCNQ2/3 activity, specifically, for example, through gene therapy. Unfortunately, this type of research is extremely expensive and complex, and viral vectors are still being explored as a delivery method of these genetic changes or genetic materials. So, but it does not mean that this has absolutely no potential. I think the potential is massive. Yeah. Now, if we don't talk about tinnitus, this principle can be noticed in different pathologies. For example, in epilepsy, the loss of K plus currents leads to hyperexcitability or hyperexcitable networks that may also alter synaptic strength in unwanted ways. Too much LTP, forming the epileptic circuits. Now, conversely, in some cognitive disorders, excessive LTD might occur in certain channels. If these channels make the neurons too hard to excite, for example. Now, basically, the ion channelopathies can be caused, diseases of ion channels, for example. They often have a downstream effect on how plasticity is gated. And in the case of tinnitus, as shown in the research I mentioned before by Thanos, it's a very good demonstration how fine-tuning of ion channels maintains the balance between LTP and LTD. Now, what does this mean for the Susan Shore device? Nothing really, actually. So thankfully, Susan Shore had discovered some timings on an in vitro level, meaning like in a test tube, that do work to drive LTD in tinnitus patients. But everything that I've mentioned before shows the complexity of timing rules and how one timing rule will not work for everybody, because everybody has a different level of ion channel dysfunction. And it's really important to keep this in mind when using a bisensory or bimodal stimulation treatment. Is there a way to do this? Potentially, yes. Of course, it would require a sample for example, from the person's dorsal cochlear nucleus, and related pathways, which could potentially kill the patient. So, in my opinion, I think, at least what I've been doing, is I've been running some computational models based on people's experiences at different levels of hyperexcitability and hypersynchrony to try and find the perfect timing for each person. But unfortunately, right now, the best we can do, due to what I mentioned before, the calcium channel dynamics, the potassium channel dynamics, and also the conduction delay, for example, which is different for each person, which can lead to a 1 to 2, maybe even up to 5 or more milliseconds of delay of signals to reach the DCN synapses, can also be a factor. So all of this put together makes the bisensory bimodal stimulation treatment a very, very experimental process, because there's a lot of guesswork involved. All right, well, that's basically everything I wanted to say for today. If you have any more questions or you'd like to discuss anything, join my Discord server. And if any of you guys have any questions regarding my trials, yes, they have started. And yes, people have received their devices already. And data is being collected, so keep your eyes open for that. That's about it. Peace.